Hi, everyone. Thank you for coming to the IICD seminar today. We are very honored to host Dr. Andrea Sottoriva from the Human Technopole. Andrea, I'm not going to go through his bio except say that he got his PhD as a student of Simon. So we are kind of very happy to have him here. And I think Simon is also very happy to have him here. Yeah, awesome guy, does awesome science, a lot of combination of theory, physics, computational biology, math, and a lot of work on cancer evolution. So anyone that's interested, you should pay close attention to Andrea's talk and bug him later as much as you want. So thanks, Andrea.
All right. Thank you very much, Sonia, for the very kind introduction. I'll start. So this is the title of my talk. It's very long, but I'll try to make it more entertaining than it looks. I'll start with a quote from a film. This is an award-winning 1959 Mario Monicelli, The Great War. It's a movie about World War I, so which is very relevant for the times we are living now. But I thought that the trailer quote was very in line with my talk. It says, this movie is an alternating sequence of tragedy and comedy. It's always in life. So I thought this really applies to my talk. It's a sequence of tragedies and comedies that I'm going to drive you through. So this is where I work now. I used to be in London for a few years at the Institute of Cancer Research. Then I moved to the Life Science Institute in Milan, which is a new institute we have been building on. That's where it is. These are the funding bodies that funded the research that I'm going to present today. Okay, this is my main topic of research, which is cancer evolution. So for a very long time, clinicians and scientists have known that tumors change over time. They change morphologically. You know, they observe premalignant lesions like a melanoma, which is in premalignant form, some sort of mole in your skin. Okay, and they observe that they transform from benign to malignant, from malignant to metastatic, and finally they develop resistance to treatment. But it is only relatively recently, in the last 20 years, that people have really managed to demonstrate that the underlying process behind this change is a Darwinian one. So the same rules that apply to the evolution of species also apply to the evolution of cancer, so the transformation of malignancies. And the main topic of my lab is trying to measure this complicated process in humans, so in patients, and trying to predict the course of the disease. That's the big question. Is cancer evolution, which is evolution on a fast track in humans, measurable and therefore predictable? And I can't have a talk here without introducing the fantastic work that Simon and his collaborators, particularly Dara Shibata, did before what is now fashionable cancer evolution. So they used to measure the evolution of tumors by looking at the length of microsatellites in the genome of cancer cells. And basically, we're among the first that apply evolutionary theory of classical population genetics. So the same piece of theory that is used in molecular evolution to understand the evolution and migration of humans applied to cancer cells. This is a fantastic paper from 2000s in PNAS, where they demonstrated, they reconstructed, right, so the number of cell divisions that the cancer had to go through to present it as a full-blown tumor. So the idea is using molecular markers like they were rings in a tree trunk to understand the age of a tumor, how it grew, how it spread, etc. So this is foundational work that really showed how they were doing cancer evolution way before it was cool. All right, a lot of things, therefore, I learned during my PhD with Simon. I learned how to do data modeling with population genetics in cancer. Other things I learned, as you do in a PhD, is you grow the PhD teaches you how to grow as a human being. And other things I learned was that if you take 1 part freshly squeezed lime juice, and then you add half a part of agave syrup, and then half a part of water, and 2 parts of tequila blanco, not Jose Cuervo. I never understood why, but this was what Simon told me, never Jose Cuervo. and you have a lot of ice, you make a perfect margarita. This is what I learned. I learned this precisely during my thesis correction. That was 2010. But then after this recipe that he showed me in first person how to do it in his house in Cambridge, he told me, I remember, son, the scaling law. And you told me this thing. And you told me, one is good, makes you great, makes you happy. Two is, borderline, it's great, but watch out, because three, you're totally knocked out. So never get hit. That's what he told me. This is a true story. Okay, so I learned how to do data modeling in cancer and how to make perfect margaritas. These are really, I'm not now. I look like I am, I'm trying to gain some of your favors, but these are the best margaritas I ever made. I didn't like margaritas before I learned how to make them from you. And they are really, I made them ever since. Anyway, all right. I said the most important thing I have to say in my talk. Now we can go back to cancer evolution. Is cancer evolution measurable? So more than 10 years ago, we started looking at colorectal cancers and full-blown malignancies in the colon. Colorectal cancers are very, let's say, archetypal type of tumors, solid tumors to examine because they are easily accessible as solid tumors. When you have colorectal cancer, half of your colon also get resected. So you can open the colon and observe the malignancy. You can sample it very well spatially because you just have access to the tumor material. And back then, with Dara Shibata, Christina Curtis at USC, what we did was sampling multiple of these so-called colorectal cancer crypts or glands. So we exploit the fact that tumors are still organized in these glandular structures, many of them that resemble the normal colon. They are like these invagination crypt-like structures that are a bit bigger than normal colon crypts. So they could be resected and isolated, okay, from different regions of the tumor. And we use old school copy number arrays and some other NGS techniques and methylation molecular clocks to sort of collect molecular data from these cancers. What we realized was, forget about this, the title, is actually that a lot of the complexity in the data that we were observing was actually originating for relatively simple rules. Okay, so when you look at the data, they look really complicated. They look completely ununderstandable. Okay, you see a lot of molecular heterogeneity. You think that the process behind this complexity must be complex. Okay, so we expected a sort of the dynamics of this growth to be extremely complicated. Hundreds of different clones with hundreds of different selection coefficients, all growing and moving and doing different things. But it turns out that with relatively simple models, computational spatial models of cancer growth, you could explain quite a lot of the data. So the complexity was originated by the stochastic branching process of cells dividing, but actually relatively simple rules, okay, with just cells mutating and expanding would explain quite a lot of the data. So all of a sudden, the apparent complexity could be modeled with relatively simple mathematical models, and I'll come back to that later. That's the main take-home message. After that, we spent 10 years, basically, trying to develop computational and mathematical methods based on population genetics. Okay, population genetics is this big field in mathematics that deals with molecular evolution and the genetics of populations. In this case, applied to cancer cells. Cancer cells are basically asexually dividing populations, okay, where, of course, there is no recombination because the whole genome is linkage disequilibrium, so it's copied over to the next generation. There are other complicated things of the data, more complicated than species. That's because chromosomal changes can happen in cancer, so you have to model that. And we were interested in measuring evolutionary forces in these branching processes where basically sometimes you have mutations, some mutations may confer a selective advantage, so the clone grows more than the rest. Okay, but back then, of course, in the previous paper, I showed you we had multi-region spatial molecular profiling, but that was not very common. Okay, there were very, very few multi-region sequencing papers. Most of the so-called intratumor heterogeneity data we had access to were basically deep NGS data, where basically what you do is you take a chunk of a tumor, you mix everything together, and you sequence the DNA of the cancer cells many, many times. So what you get is a long list of mutations in your tumor, and also not just the mutations, but the proportion of the sequencing reads with the mutation versus the non-mutant sequencing reads, which you can plot in a histogram for the allelic frequency, which in the old school population genetics called sex frequency spectrum. And you can plot the histogram of the frequencies of these mutations. So how many mutations are at 50% in your cancer? 50% probably means that every single cancer cell has it if it's in a diploid site, and how many mutations are at different other frequencies, etc. Okay, and these histogram distributions contain information about this process. Okay, for example, in this case, you see a nice so-called clonal cluster of mutations, mutation high frequency, in this case on 50% allelic frequency. That means they are in every cancer cell in one of the two chromosomes. So these are mutations are in, for example, this is here, and every cell has them in one of the two chromosomes. But then you see also a lot of mutations at lower frequency here. And you may see different bumps in the distribution, which means different things, which I'm not going to deal too much with at the moment. But what we did was basically spending a lot of time designing computational and mathematical models that can reproduce this data, and therefore basically measure different evolutionary forces from these distributions. And that is because the laws of biology do determine the distribution of these mutational frequencies. So if there is a selected subclone that's growing here, then you have certain structures in this variant allelic frequency spectrums, for example, these clusters over here. So you see selection, okay, of mutations in expanding clones that takes a higher frequency mutation that should have been at low frequency. So you see higher frequency mutations that you expect by chance. These are probably under selection, etc., etc., etc. Then we combine these computational methods where they were all about forward simulations of frequencies of mutations. So we were simulating a lot of times cells dividing, mutating, and in standard computational simulation. It was not scaling very well. These simulations are relatively hard to fit. You have to use Bayesian inference and do a lot of simulations to fit your parameters. So we try to bridge the two distinct worlds that were common back then in analyzing variant allelic frequency spectrum. One was what we were doing with basically simulations, and the other one was machine learning based clustering, which is called basically Dirichlet non-parametric clustering, where basically you say, okay, I don't care what these distributions structures really mean. You just, I know they are, a mixture of distributions. So it turns out that basically when you combine these sort of model-based simulations of allelic frequencies with non-parametric Dirichlet clustering, you can basically much quicker identify these different structures. And what we could do is separate these clusters of mutations of high frequency, which contain mutations driven by selection. Okay, these are clonal mutations, and this is, for example, a subclonal mutation. Sorry, subclonal cluster of mutations at higher frequency that arose at this frequency because of selection. They could be combined with this typical power law distribution of this branching process, where more and more mutations arise at lower and lower frequencies in 1/x squared tail, which is a known law in population genetics. So you can basically take this data and cluster them with this type of methods, which we call Mobster, and basically extract measurements of this of the evolutionary process from these distributions, because the height of this histogram is proportional to how old this clone that is expanding is. The frequency also, place where it is in this histogram, depends on the selection coefficient, and the steepness of this curve depends on the mutation rate. So you can extract evolutionary parameters from this distribution. You can do the same in space. Okay, so you can sample different regions of the same tumor. That's what we did, again, exploiting single gland isolation. This is a colorectal cancer with adenomas and carcinomas. You can identify different regions of the big blown malignancy there. You can sequence the genomes of different crypts, okay, which are this one. Each one of these leaves in this tree is a single crypt, which are rather clonal, tiny clones, basically about a few thousand cells big. You can reconstruct its phylogenetic tree, like a tiny tree, okay, in different algorithms. And you see, for example, look at this tree. This is real data, right? what do you see? You observe that crypts that come from the same region, which are marked with the same color, they are close together. So there is some spatial structure here. So crypts that are here nearby are also genetically similar. Okay. You see here a longer branch, intermediate branch in this tree, which suggests a clonal expansion. In fact, in this branch, there is a mutation in KRAS, which is a non-oncogene. And in fact, what you see is you see a clonal expansion. Okay, so you detect the effect of selection here. And then what you do to measure parameters from this tree is you do a forward simulation. So you find the right spatial simulation that basically is able to recapitulate the tree that you observe here. This is a real tree. This is a simulation. Okay. So you can simulate a tree when the parameters fit the data, you get like a tree that is very similar to the real tree, and therefore you can measure selection coefficients and mutation rates, etc. So in conclusion, the laws of biology and of evolution determine the spatial distribution of the mutations in the cancer. So we were using the distribution of the frequencies of the mutations and how they are distributed in space to extract evolutionary parameters to measure this process. Okay. So to cut the story short, cancer evolution is a measurable process. Okay. But is it predictable? Or can you make useful predictions in cancer evolution that can be exploited in the clinic? There are different ways to do it. Okay. Back then, we thought, well, we know that cancer evolution is driven by often a sequence of genetic alterations. Okay, if you take the simplest example of colorectal cancer, very often these are initiated by an APC mutation, and a KRAS mutation, and a P53 mutation. So do different evolutionary trajectories or sequences of these events lead to different evolution of the tumor? So if you take different patients that you observe being different genetically, okay, you may argue that some subgroups of patients have similar evolutionary trajectories. Here you have the yellow and the blue. Okay. So the hypothesis was, can we reconstruct these recurrent evolutionary trajectories, and are they useful? Okay, are they prognostic, for example? So if you do multi-region sequencing in different patients and you reconstruct their phylogenetic history because of noise, because of the stochastic process behind this generation of mutations, you usually get every time a different tree. Okay. So you don't see a lot of recurrent in these patients. Even if they have the same mutations, very often the sequences of these mutations on the tree is a bit all over the place. So what you can do is, instead of reconstructing with algorithms such as maximum likelihood, okay, in each patient, a different phylogenetic tree, you use a machine learning technique called transfer learning, where instead of doing, I think, one patient at a time, you look at the big picture, you look at different patients, okay, and try to find recurrences in different groups of trees for every patient. So when you try to reconstruct the phylogenetic tree of one patient, okay, very often there are many phylogenetic trees that fit your data. Maybe not as well as the one picked by your maximum likelihood algorithm, but you get a lot of trees, okay, that maybe are not the best tree, but they are the second best tree or the third best tree. Of course, if you always take the best tree, you are going to influence by the stochastic effects in a single patient. In this case, what we wanted to do is to look at many of the best solutions there are for every patient and basically try to optimize for recurrent evolutionary trajectories where sequences of events in the tree may be repeated in other patients. Okay, so sometimes you basically swap some of the branches of the trees to optimize recurrent evolutionary trajectories across patients. So all of a sudden, before you had different evolutionary trajectories for every patient because of stochasticity, because of noise. Instead, in this case, because you apply transfer learning, you pick different suboptimal trees for a patient that, however, they maximize the likelihood of groups of patients to find recurrent evolutionary trajectories. In this case, for example, that's an example in breast cancer, you find that basically you go down so some decision tree when you accumulate mutations in breast cancer, and therefore different evolutionary trajectories also, in fact, corresponded to different prognostic to survival, basically. So different evolutionary trajectories indicate different progression-free survival. All right. We did something similar with multi-region sequencing in prostate cancer. So in prostate cancer is a great cancer type, even more than colorectal, to look for to make predictions about prognosis, because it's a cancer that is usually diagnosed much earlier on than, it's usually diagnosed relatively early. People go on for a long time with prostate cancer. Some people do very badly and some people do very well. So it's very hard to prognosticate people. It's a cancer that is diagnosed through multi-region biopsies. So if people suspect that the patient has prostate cancer, they do a transrectal multi-needle biopsy. So we had cohorts of patients where I was before in London, where we had a lot of samples from the same patient already. So these are like needle biopsies, transrectal needle biopsies. Prostate cancer of different regions of the prostate. So you have different regional samples, okay, that we could profile with for genome sequencing to look at copy number alteration. So we have the genetic heterogeneity of a given patient, and we also combine it with digital histopathology. So we use basically deep learning to identify different Gleason grade regions. Gleason is basically a measure of how messed up your cells are. This Gleason is the measurement by which a histopathologist determines how far advanced is your prostate cancer. And the higher the Gleason, the worse is the morphology of your cells, the more untidy they are. It's a typical prognostic factor in prostate cancer. So what we did was basically analyze all this data in light of this automatic Gleason scoring with deep learning, together with the genetic profiling of each biopsy. And if you measure the heterogeneity in terms of divergence of at the genetic level and morphological level, okay, of these cancers, you can identify groups of patients that are predicted to relapse after 10 years. This was a great cohort because we had more than 100 patients for which we had multi-region biopsy 15 years before. This patient was followed over time for 10 to 12 years. So we had very long-term follow-up. And these were all like tissue blocks that were saved in histology for a long time. So we could test whether this genetic and morphological heterogeneity was really prognostic because we had long-term follow-up. And indeed they are. So in conclusion, of this first part is cancer evolution in humans is a measurable process. Okay, that if you combine data plus theory, you can actually deconvolute, you can understand it. We can, through population genetics, measure evolutionary forces, positive selection, immune-mediated negative selection. I don't discuss it today, but you can do it, and mutation rates in patients. And some of these evolutionary measurements do predict clinical outcome. And I showed you how they predict prognosis. Now, this is, on one hand, it helps highlighting the actual explanatory power of evolution when applied to cancer, because it really shows how by understanding evolution, we understand cancer biology. In reality, that's the tragedy of this first part of my talk, is that it's really of limited clinical use. So it has been, we should admit, a bit disappointing to the clinical world, cancer evolution in some ways. Because we all expect 10 years ago that when we did all of this work of measuring all these evolutionary dynamics in cancer patients, clinicians would really know what to do about it. And rightfully, they don't. Yeah, we can predict prognosis. So what? What are we going to do to these patients? We're not going to start putting them in screening because we have, we don't have, even screening is changing screening is really complicated. Okay. For example, not screening some patients because we think they are going to do well requires a prospective clinical trial that demonstrates that lack of screening is not detrimental to patients. So it's extremely, in general, prognostic biomarkers are really difficult to implement in the clinic. And then we had other problems. And that is that very often we were observing these clonal expansions. Okay, so these selective forces that would expand populations. You see expansion in a phylogenetic tree or increase in frequency. You see bumps in the frequency spectrum, but they do not contain any known drivers. People have looked for driver events in the non-coding genome, but it's not been very successful, apart from very, very few cases. And therefore, we were a bit stuck with basically having to make predictions about using basically only genetic events of a process of evolution in which some of the stuff was not even measured. Okay, so we were not capturing some of the events that were driving this selection. So the question was, how much of the cancer evolution is actually not genetic but epigenetic, and how much is not even evolution itself, is like non-heritable plasticity, which is not evolution. And the problem was also that a lot of population genetics theory does not tell much about epigenetic evolution, because of course in population species we are concerned about genetic material being passed, not the epigenome. All right, so we decided to create a big multi-omic dataset from colorectal cancer. That was back 2016-17 we started. So again, we did this multi-region single gland sequencing from these carcinomas and various other lesions. So you extract glands or crypts that they look like these, again, structures here. They are not single cell. You could not really do multi-omic single cell sequencing back then. Certainly not multi-omic with single cell DNA sequencing. So we exploited crypts. Crypts again are about 5 to 10,000 cells big. They are really amenable to multi-omics. You don't have enough material to do multiple assays in the same crypt. So we generated this large data set for about 1,300 crypts, where we had whole genome sequencing of individual crypts plus RNA sequencing plus ATAC sequencing, which is basically a surrogate of chromatin accessibility. And because we come from genomics, we thought, okay, let's just treat epigenetic changes, which are changes in chromatin accessibility at the promoter level, in enhancer level. Okay, let's treat them as they were mutations, even though it's not really true. So we said, okay, we call them somatic chromatin accessibility alterations. And we say that an alteration can be gained. So a piece of chromatin, imagine this was a chromatin, to be closed in the normal epithelium. So the gene is not expressed and the promoter is closed. And instead in the cancer, it could be open. Okay, so this is a gained accessibility. The promoter opens up and the gene is expressed in the cancer. Or you can have that the promoter is closed and the gene is lost in the cancer, the expression is lost, we call them lost cell. So you treat them as a binary on and off, okay, even though they are not. What it turns out is that when you do that, and you look at promoters and enhancers on known cancer driver genes, pan-cancer, not colorectal cancer driver gene, but any cancer driver gene, you find quite a lot of recurrent promoters and enhancer epigenetic changes in this list of cancer driver genes. In highly recurrent driver genes, such as CCDC6 is a tumor suppressor gene in colorectal cancer, if it recurrently closed at the promoter level in colorectal cancer. And these are very often devoid of mutations in the gene. The mutations are marked with this like purple star. Okay, so in many cases, you switch off a tumor suppressor gene just by closing the promoter without the mutation. So this is not very surprising. Right? So you find like a plethora of cancer driver genes that are changed epigenetically through their promoter or enhancer. And of course, if you compare adenomas to cancer, they have different load of these chromatin changes. So of course, the more cancers have many more of these chromatin changes as they have more mutations in copy number. And then I told you we had the epigenome, the genome, and the transcriptome. So from the genome, again, take this cancer here, from the genome, you could reconstruct the phylogenetic tree. That is this one. Use DNA to reconstruct the phylogenetic tree of the cancer. And then for every sample, you also have their transcriptome. Okay. So in this heat map here, you see basically a surrogate of a transcriptome. And then we had a question, the question was, does the phylogenetic tree control transcriptome? So is gene expression controlled by the genetics, driven by the phylogenetic tree? And you can use a tool for population genetics, which is called Pager's lambda, to determine whether certain trait, in this case a transcriptome, is actually determined by the phylogenetics or not. And it turns out that in the majority of cases, it is not. So you have things that have samples that have similar transcriptomes, okay, that are, however, very distant in the phylogenetic tree. So if you take the samples here, okay, they are really divergent. So these samples here and this here are very flat. They have nothing very divergent genetically, but their transcriptome is very similar. And in fact, guess what? They were in the same place. So if you notice here, they are both purple. These two sets of glands, okay, of crypts. They both come from the same place, but they are genetically divergent. But because they are in the same place, that is very similar, suggesting that, of course, the tumor microenvironment can drive transcriptomic profiles independently of the genetics. All right, so we started basically mapping these heritable epigenetic modifications. We know they are heritable because you find really glands that are really far apart with the same epigenomes, and they have recurrent alterations in promoters and enhancers of known cancer driver genes. So, not surprisingly, epigenetics drives cancer evolution as well. You can do an analysis. I didn't present it much yet, but you can do an analysis at transcription factor level by looking at accessibility across the genome, transcription factor, effective interesting stuff at the level of reactivation of developmental genes. A large proportion of transcriptome heterogeneity is not variable, but it's plastic. So, okay, a lot of transcriptome is not controlled by the genome or the epigenome. And then we thought, okay, this is going to be useful only if we understand it also through treatment. Okay, so the main question here was, okay, you can see epigenetic evolution in primary colorectal cancers. Does it explain some of the unexplained treatment resistance? If you take one of the cancer types for which we know most of the mechanisms of drug resistance, this is EGFR mutant lung cancer. If you give anti-EGFR to EGFR mutant lung cancers, it very often progresses with additional mutations, either in EGFR pathway or in KRAS or in other members of the pathway you target. And still, in this best case scenario, about 40% of the patients have totally unexplained resistant mechanisms. So the genetics doesn't explain this large part of drug resistance in lung cancer, and certainly in the large majority of colorectal cancer as well. Also in colorectal cancer, we see a lot of drug resistance, but a tiny minority of these cases, this is explained by genetic mutation. So we thought, is epigenetic evolution driving that as well? Okay, so we look at different cohorts. One of them was this clinical trial here, where we look at patients that got colorectal cancer patients that got anti-EGFR treatment over time, and we had like ctDNA over time. At baseline, at best response, these patients responded initially, but then progressed. Okay, so we went two different arms, both with anti-EGFR, slightly different treatments. We thought, okay, we are going to do deep whole exome sequencing in ctDNA with UMIs. UMIs allow you to detect very low frequency somatic mutations at the exome level, and we are going to find recurrent genetic mutations and progression. Because this is a typical clinical drug resistance example. You respond to treatment very well, your disease almost goes away, and then 12 months, 24 months later, you come back with a resistant tumor, and the treatment doesn't work. We did this analysis of the variant allelic frequencies where you have baseline versus progression. Okay, so you have mutation that you observe in both the samples at progression and baseline, which are C mutations that known driver events. There are some mutations that you only observe at the relapse. Okay, this one in red, you can see them here. So if you look at the progression time point, you have again this variant allelic frequency distribution. You see here the frequency is very low because the purity in ctDNA is very low. There's a lot of normal contamination, so you have to develop special assays for that. So you find in green the clonal mutation, then you have the usual suspects inside. And on top of it, you start finding these new mutations that arise at progression. And we were convinced, okay, that these new mutations in red would contain drivers of resistance. and they don't. Okay. So when you do the NDS test, you basically pull different mutations in these new mutations that arise only at resistance in all patients, okay? And you look at subset of genes or even all the genes you have, you do this DNDS, this enrichment of non-synonymous versus synonymous mutations. You should see the red dot here. If this red dot is bigger than 1, that means there are drivers inside, and it's not bigger than 1. Okay? So you don't find, even though there are these clonal expansions, okay, so there are these expanding clones that relapse, inside here there's nothing recurrent. Okay, we had like 40 patients, 50 patients. There's nothing really recurrent, nothing that explains why these patients are resistant. So the clones, something took over the population. Okay? And these patients are fully resistant to treatment, but somehow it's not genetics, okay? Because there is nothing, at least we don't think this, there is nothing different. So there is little evidence of emerging genetic mutation in that treatment. So again, we turn to epigenetics, which is very difficult to do in ctDNA. You can do it with that thing called fragmentomics. So of course you don't have tissue, right? It's circulating tumor DNA. But if the tumor DNA is the piece of DNA that is shed in your plasma was in open chromatin, it degrades very quickly. Okay? And if it is in closed chromatin, then it gets slower. So if you look at the coverage of certain regions that were in open chromatin in the tumor, then in the ctDNA you see basically in the coverage. So you can basically extract epigenetic profiles from the fragment size of circulating tumor DNA. And then you can compare progression versus baseline, which is the 14 meters. And you start seeing changes all of a sudden. You see a lot of changes, some changes in chromatin remodeling genes. So you start observing an epigenetic evolution process occurring under treatment, okay, that we think, may explain rather disease. In reality, there is a limit of what you can do with circulating tumor DNA because you can only see broad changes in transcription factor binding sites. That's because you cannot really do a pair site analysis, okay, because the signal is very, very weak. So we really had to go to tissues, okay, so go back from circulating DNA to DNA, which we thought was the way forward to understand drug resistance, because normally you cannot rebiopsy patients. Drug resistance is very hard to do because patients go through several lines of treatment. It's very hard to convince doctors to rebiopsy patients that have failed first, second, and third line treatment. So it's really rare to have paired pre and post treatment biopsies. And so we thought we're going to do everything in ctDNA. That was like 5-6 years ago. But we ended up that actually, because of a lot of the resistance was driven by epigenetics, in the end, you can do some fragmentomics, but you have to go back to tissues to understand really what happens. So we set up a tissue collection effort in neoadjuvant and window of opportunity clinical trials. So window of opportunity clinical trials are those trials that we run at National Cancer Institute, where basically you take early patients that are like stage 2, stage 3 patients that are resectable, okay? You can send them to surgery with potentially curative intent, okay? Late stage patients, very often they do not even go to surgery. So if you have a lot of metastasis, they just give you chemo and best of luck. And they're never going to rebiopsy you because things are not going very well anyway. So these patients can go to resection. So when they present in histology, sorry, in diagnosis, we take a biopsy. Okay, so the diagnostic biopsy is saved for research, the biopsy. So we freeze the biopsy, and we also use another biopsy to derive patient-derived organoids. These patients get put into this window of opportunity clinical trial depending on the molecular profile they have in the biopsy. So in this case, in colorectal cancer, they may have a KRAS G12D mutation, G12C mutation, for which there are drug targets. They may be hypermutant, MSI high, and they go on to do immunotherapy. So the idea is that between the biopsy and the surgery, you can introduce a treatment. Okay. And the nice thing in this case is that this treatment is basically a monotherapy. Normally, patients get like combination of therapy. It's very hard to make sense to samples that have undergone several lines of treatment. In this window of opportunity trial, we get treatment naive patients for which we take the biopsy before treatment, then they get given this window of opportunity treatment, which could be immunotherapy, could be anti-GFR therapy, panitumumab, could be anti-KRAS G12C therapy. And then they go to surgery 4 weeks later, and we retake material during surgery. So we have pre-treatment, treatment-naive samples, and post-treatment samples. And we do this. So what we do in this case, we do single-cell multiome, so standard 10x RNA and chromatin accessibility at single-cell level of these biopsies. We observe the same thing we did in crypts. So you observe a lot of recurrent chromatin accessibility alterations in promoter enhancers, in non-cancer driver genes, which is and the incidence is inversely proportional of the genetic mutation. So you have genes that are recurrently mutated at the genetic level, p53 KRAS, and genes that are recurrently mutated at the epigenetic level. You don't have time to go through all of them. You find recurrent epigenetic changes in the normal adjacent cells as well. I'll talk a bit more tomorrow in another talk about this. But I don't have time. So what we did was, we started then comparing the biopsy and the surgery of patients that have undergone a single line of treatment. Okay, in this case, I give you a couple of examples, a couple of patients that have undergone panitumumab, which is anti-GFR. Okay, the biopsy. So what we did was we did differential chromatin accessibility at the surgery versus biopsy, so pre versus post treatment. Okay, and all of a sudden here, where there are no genetic alterations that could be, you do all genome sequencing, these two samples are identical. You start seeing recurrent changes at particularly in promoters of certain recurrent driver genes, okay, that are, as I said, recurrent. So you start seeing recurrent changes in non-driver genes, okay, some of them interpretable in these patients that have undergone anti-GFR therapy. If you look at the transcription factor binding site accessibility, you see something that is quite obvious if you think about it. So you see a lot of suppression of Wnt signaling. Okay, that's obvious because you give the drug that suppresses Wnt signaling, which is anti-GFR. Okay, so you see downregulation of LEF1, TCF7, etc. And then at the same time, you see reactivation of these anti-apoptotic signals and pro-survival signals driven by these transcription factors that you see here. So basically, these non-responders, okay, these are patients that receive anti-GFR therapy but did not respond. You see the effect of the therapy, it switches off the GFR, okay, but nevertheless, the cells don't die. You start seeing these resistant drivers coming up, driven by the epigenetic rewiring at this transcription factor level. This is another couple of patients that got botensilimab, is a CTLA-4 inhibitor. So this is an immunodrug. And again, you see a recurrent, two patients in this case that were collecting more data analysis. You see here that recurrent, it's recurrent epigenetic changes, pre and post treatment, and the same at the transcription factor binding site. So you basically start seeing recurrently downregulated transcription factors in post-treatment. All right, summary. Epigenome cancer evolution is evident. Somatic changes at the epigenetic level are really recurrent and are stable. And a lot of epigenetic changes occur in the absence of a mutation. And I didn't have a lot of time to talk about it, but epigenetic rewiring also happens in the adjacent normal cells and the tumor microenvironment, and you observe it with this multi-omic approach. I don't think I have. Do I have 5? No.
Yeah.
Okay. In the last part, I know it's a lot of stuff, but I'll go through it very quickly, which is probably some of the more interesting from the modeling point of view. Everything I showed so far is in patients. In patients, you can do modeling. You can look at bioinformatics, look at recurrent events, and try to make sense to them. But in parallel, what we have been doing in the last 10 years is to build experimental models that we could use to test the mathematical models that we have. So because we wanted initially to stay away from cell lines, and we adopted these patient-derived organoid models, which are becoming quite established now. So we routinely derive gastrointestinal organoids from our cancer samples. And the nice thing of colorectal cancer is, again, that because we receive colectomies, which is half of your colon, we can derive normal colon organoids and cancer colon organoids from the same patient. So we have a live biobank of about 150 patient-derived organoids from about 250 patients. So we have a lot of these model systems. They are not perfect. They recreate some of the structure of the colon. They make crypt-like structures. They have different cell types inside. You see Paneth cells in colorectal organoids. You see transit amplifying cells like in the colon. Okay, so they don't have an immune system. They don't have stromal cells. You have to make the cultures. You have to artificially recombine them. These are only epithelial cells. But they are, we think they are useful model systems to study drug resistance. And back then, together with the collaborator at the IICD, Nicola Valeri, we did show in co-clinical trials that if a patient responds to, in this case, chemotherapy, the organoid also responds to chemotherapy and vice versa. So we thought they were like a good model system. So we use them routinely as models of experimental evolution. Now, if you want to emulate in the lab what happens in the patient, you need a model that is patient-derived in the sense that it has to recapitulate the genetic and epigenetic features of the tissue of origin, okay. But at the same time, a model that is controllable, is easy to use, but also that you can expose to drugs for a long time, okay. And therefore, standard drug screening was not something we were looking for. In drug screening, you take the organoid or whatever model system and you do like a 7-day assay, okay, and you check which drugs respond to treatment or not. But there you don't see resistance, right? The idea of resistance is that you expose your cells to drugs and you wait and keep on exposing them like a patient, right? And 4 weeks, 5 weeks, or 3 months or 5 months later, you get resistance out, okay? So you want to simulate resistance in your experimental setting. So that's what we wanted to do. So that's what we did. So we took patient-derived organoids. We wanted to track their evolution over time. Now, to do that, we use off-the-shelf lentiviral barcoding. You can just buy random barcodes and basically infect cells with tens of thousands or hundreds of thousands of random barcodes. You plate them in these matrix domes in 3D, okay, and you give long-term exposures to drugs. In this case, we had sequences of drugs of different target inhibitors. Okay, so we give like 45 days of a certain drug. Okay, we wait for a lot of cells to die. Okay, then we suspended the drug. We wait for the resistant, persistent cells to grow back. So we had sequences of drugs over long periods of time. Okay, so 4 to 6 months evolution of these. In these organoid models without replating. That was also another thing we wanted to avoid. Normally, when you try to induce drug resistance in cell lines, you keep on replating them with higher and higher dose. We had very high dose at the beginning. We kept it at very high dose without replating. Replating is a difficult thing to model mathematically because you are saying, okay, I'm going to take off the cells from my matricial dome and replate them back. We wanted to avoid that. And so that's what we did. Then we had different drugs that we wanted to test on these organoids, and we also have different sequences of drugs. We were interested in understanding mechanisms of resistance, but also understanding if there were certain sequences of drugs that work better than others. So do different drugs select different clones? That would be a good news because that means you can combine drugs. If you have drugs that all select for the same clone, basically you lose control very quickly. So we could test sequences of drugs and track the clones at the same time. I remind you that we infected cells with random lentiviral barcodes. Imagine 10,000 different barcodes, 1 barcode per cell. Okay, and then what you do is you plate these organoids in different replicas. Okay, the distribution of barcodes is the same for every replica. Then you give the drug, and then you reprofile the barcodes at the end. What you expect is that you start from thousands and thousands of different barcodes that you find online at the beginning, and you end up with less barcodes, okay? And if you end up with less barcodes, I mean you had a clonal expansion, okay? A few cells took over the population. So look at this, you have thousands of barcodes, here you have only one barcode. And the interesting thing you observe is that when you look at the distribution of barcodes across replicas, you find exactly the same barcodes coming up, okay? That means that selection occurred on pre-existing subpopulations of cells. So if you have 3 replicas with tens of thousands of different barcodes, the same barcodes across replicas, okay, you may have that drug resistance arises because there is while you're giving treatment, a random mutation occurs. If that happens, you expect different barcodes in different replicas because this mutation occurs in a random lineage, okay? You may have many other mechanisms of resistance. In the classical Darwinian evolution, one subclone already was resistant in the first place and expanded in the 3 replicas independently, and therefore you have the same barcode across 3 replicas. This is how you test for pre-existing resistance. So here you have the parental line with 3 replicas, and then you have different drugs. So these are AKT drugs, with target AKT, AKT inhibitors, and then there's a MEK inhibitor over here, there's an mTOR inhibitor. So what you observe is highly repeatable evolution. You always select the same subclones, okay, but different drugs select for different subclones, which was quite interesting. We really like that. And at the same time, what we were doing, which was quite cool, was every 3 days you have to change the media. So you have to change the food that these organoids have, this liquid that is on top. Normally you throw it in the bin. So instead of throwing it in the bin, we thought to spin it down and extract DNA from it, and therefore extract the lentiviral barcode pieces of the DNA with a PCR. With the idea is that all the cells that remain on the bottom are the live cells, and the floating DNA comes from the dead cells. So we could use it for the modeling. And the nice thing is that this works like circulating tumor DNA. So if you look at this long diagram, which lasts like 150 days, okay, we only took solid samples. These samples that are here, they come from like 3 or 4 time points, okay? Because when you really take the cells out. But every 3 days, we could collect these floating barcodes, which really mimic the underlying evolution of the cells. So if you look at here, for example, this sample here is actually this solid sample here. So you see the proportion of green and blue are the same that you see over here, okay? So you see the dynamics here, and at the end, that's the same. So the proportion of blue here, the same. So these are all samples taken from like some sort of circulating tumor DNA of your tissue culture. Now, it sounds counterintuitive that basically the dead cell look like the live cells. Okay. In reality, if you think about it, at the beginning, when with the drug you kill a lot of cells, you have a lot of random barcodes, okay, in the dead cells. But after a while, when the population stabilizes and you have a dominant clone, you only see the turnover of that dominant clone. So the only barcode you can sample is the one of the dominant clone. So this is very useful because from a modeling perspective, you have basically a very dense longitudinal sampling of the barcodes. All right. So Darwinian selection for pre-existing heritable clones. Okay, so highly repeatable evolution, you select always for the same pre-existing clones, and we thought, okay, they must have some genetic alterations inside that explain why they are resistant to MET inhibitors and AKT inhibitors and whatever. All right, so we thought, okay, because we have this is single cell RNA-seq from which we extracted lentiviral barcode. Okay, so you think, okay, if this is the resistant clone, the blue one is resistant to MET inhibitors, okay, MET inhibitors. Okay, that means that the RNA-seq, the transcriptome of this clone must be very different than any other clone over here. Okay. All right, let me see what it is. It's not true. So you take RNA-seq, okay. This is after treatment. You suspended the drug, okay, and you let the tumor regrow, okay. You do RNA-seq, and you, in this case, so these colors, they are this is an analysis based on archetypes. Again, I'm trying to discuss it, but when you do, when you do single cell RNA-seq and you of a single epithelial population. It's not like when you take single cell RNA-seq from a tumor where you have T cells over here, macrophages over here. When you have a single population, most of the time you see a blob. Okay. So doing clustering on single cell RNA-seq is not very useful. You do a thing called archetypal analysis, okay, which looks at extreme cellular programs. Okay. It allows you to basically deconvolute the cellular programs that are in your single cell analysis. I didn't have time to go through it. But if you basically summarize your RNA-seq into these archetypes, okay, look at this, you don't see any difference in this archetypal composition. Archetypes, you can imagine them as combination of cellular programs, as I said before. So they are a summary of the cellular programs that are being used at any given moment by your cells. They are a surrogate of some sort of summary statistic of your data. When you look at the regrowth population, okay, you see this sort of distribution of archetypes is basically the same as it was before. Okay, so when you see when you look at the RNA-seq under the drug, when you're giving the drug, of course, the transcriptome is totally different. You are giving a drug, the drug is changing the transcriptome. When you suspend the drug, the transcriptome goes back to what it was at the beginning. Even worse, we thought, okay, I understand that there is like sort of bouncing back of the transcriptomic profile, but if this blue thing was selected, then the blue thing that was here there was a blue clone, it's tiny, right? But you can extract it from this population. If you look at these few cells with the blue markers here, they must have been different from the rest because it has been selected, right? So you thought that the transcriptome is different. Not even that is different. Okay, so you see changes under the drug, it bounces back. But when you look at the parental clone, the blue parental clone, okay, it was here. The transcriptome of this blue clone in the parental one at the beginning was identical to the rest of the cells. So why was this selected? Why would you select this? It looks identical to this. So why is this selected by treatment and the other ones are not? When you actually look at the epigenome of this clone, it is different from the rest. You see it here. So the epigenome of this blue clone is the same as the regrowth clone. Okay, so these have the same barcode and in fact have the same epigenome. Okay, but it's different from the rest of the population. Okay. So evidently the epigenome is different, that's what the memory is, but it's not the same as saying what genes you actually express at any given time, what type of transcriptomic profile you have. So to cut the story short, you evidently have DNA mutations, copy number alterations, and epigenetic modifications, which represent your cellular memory. Okay, but from any given, let's say, point in the fitness landscape that is determined by this memory, okay, you can actually express a multitude of phenotypes of transcriptional profiles. So you have a lot of transcriptional profiles that you can express in different circumstances from this point in the fitness landscape. That's what you observe, okay? You observe different transcriptional programs, but the memory is actually in these epigenetic changes, and that's what you really select for. Okay? So even though you observe things here, actually the memory is over there. All right. High convergence, and then distinct clones start up to different drugs, and then you have different adaptation mechanisms manifesting the same tumor. Okay, all right, I'll stop there because I think we finished time. I wanted to thank everybody in my lab in London, which started all this endeavor, and everybody in my team. Yeah, the retreats are different venues, but anyway, thank you very much, and happy to take any questions. Yes.
So, question regarding the model. So, when only considering mutation, then we use the process model, right?
Sorry, say it again.
So, for only concern mutations, when we use rich process to model, right? So, are there any models that you know that has been successful incorporating epigenetics and other operations?
So, yeah, that's an excellent question. The problem, yeah, so mutations are relatively easy to model. right? And you model mutations as a Poisson process, accumulation of a variable, because it's like a random error in your thermodynamical machinery, right?
Yeah.
Epigenetic alterations are CPG alterations, single CPGs, you can model them as that. But how do you go from a non-methylated promoter to a fully methylated promoter? That I don't know. It's an open question, how you model them. Do they happen in one go? There are some papers showing that, for example, if you have a deamination at the CPG site, okay, the error correction may strip of methylation the whole promoter. So, you know, the DNA repair sometimes creates these like quick demethylation events. So that may happen in one go, but the problem is, you know, the mutational process is not. At the moment, I would model it naively as a single event. But I don't know much better than that.
I see, okay.
So when you show the increase in the VAF, the time, in CCD and IM, has it been corrected for coming up to these?
Yeah, so, well, so the copy number correction is DNA is a bit hard because the purity is very low. What you do see is you recurrently, so what you do is you make sure, so well, you make sure you don't see new copy number events, okay? And you, so let me reformulate the answer better. When you see different bumps in the parental frequency distributions arising, and you suspect it's copy number, you can plot where these mutations are in the genome. If they cluster in a chromosome, then it's copy number. You always have to check the history. We were more for the purity. Okay, so you detect them now because the purity is high, so that you have to normalize for people. But normally, between progression and baseline, the purity is pretty similar. Okay, the problem is in intermediate time points is much lower because these responders, so they respond frequently. The node goes pretty low frequency, then it comes back. So it's not copy number. They don't have really even. We were looking at recurrent copy numbers in that relapse. It's 50 patients. It's a bit surprised. Whereas when you look at the fragmentomics, you do see quite stuff going down. So there are several transcription factors that change their accessibility. There is a limit to what you can do.
So I had a question for that last story. So when you had, you clearly have clonal selection, for some pre-existing variation. Do you see differences in archetypes or transcriptional programs upon drug?
Yes.
You do. Okay.
Once the drug is
that are clone specific, I guess, but
that are clone specific.
for the expanded clone versus the Russell.
I don't recall exactly free. So you look at the, well, it's your point. I don't know if we look into that. But I have to say that the problem of, so having the transcriptomes under the drug is informative for the effect of the drug, but they are overwhelmingly driven by the actual drug itself. So I don't, we could check, but I would say that their variation is minimal with respect to enormous effects of the drug. I don't know if I explained myself correctly. So the drug will completely change their
But I guess I was thinking if you have, a subclone that at some point gained a mutation, that doesn't seem like that was the case, but let's say that was the case where you had one mutation, that you do expect that under pressure, the lack of that one target would maybe, maybe you'd be seeing off-target effects, but not on target. And
Yeah,
that's
let's do. Maybe you should. Let's do one. We should check that. Yeah. And the caveat also is that we have very few cells under the drug because we give like IC90, so we get like these dead semi-live cells, 10%, 5% of the population, and so we have we just don't have so many cells. So I don't know if we can capture so many unless it's an expanded clone. you don't capture many rare clones, but still you would argue, yeah, we could compare that clone versus anything else to see if under the drug it was rewired. We should check it.
Another question, so, very beginning of the presentation, you showed that, there's a Big Bang theory for cancer evolution, right? So that means, in a very short, period of time, you have a lot of permutations, right? And it will come to four clones, right?
Yeah, so the interpretability, originally the model was basically a model with very weak selection. Okay, so at some point you have an initiating event. Whether the mutation rate is higher at the beginning than later, Darrell Shibata is convinced that at the beginning you have higher mutation rate, but I never understood what the mechanism is. Okay, you certainly have an event that triggers a big clone expansion, followed by very weak selection, if no selection at all. Okay, there you have that in few branches, cell divisions, you generate a lot of variation.
Yes.
Because mutation rate is quite high.
Yeah.
Log out. That variation is fixed basically in the population structure. So the first few cell divisions, okay, you observe it, it basically freezes in the cancer expansion. Okay, in the simplest case scenario, 50% of the cells have the mutation in the first division, and 25% of the cells have the mutation in the second division, etc. So you see this, the effects of the very few cell divisions very later, where you can't sample it. And if there is any cell mixing very early on, that's also you see it very fast. So the mixing that you see, sometimes you see like genetically divergent cells mixed up in the same region. If you simulate it, you also realize that it comes from mixing in the first subdivisions. Basically, the first subdivisions freeze the structure. That was the original idea. So if you are just weak selection and cell motility, then you get this big bang sort of.
Yeah. But when you are doing the cell frequency spectral analysis, that one does not appear through the visual.
So you say
The cell frequency spectrum. Basically, we have a version of cell is the frequency spectrum, right? With very little. So that one does not come from a Big Bang theory.
yeah, because you have like one event and then you basically have a branching process with no selection, where you see the effects of the first few mutations. So in the power law distribution is day 1 over squared day. Right, these are really the first few divisions, maybe 10 divisions. Okay, then you don't see them anymore. So you see these effects of these first few divisions, which you see across the life of the tumor. It doesn't matter when you sample it. Even if you wait 1,000 years and you resequence the tumor, you will see exactly the same frequency spectrum, the first few divisions. Because the others are too rare. Right, that's fine. Is that what you mean?
I'm saying that the model for you to build a cell frequency spectrum is basically from a branching evolution point of view, not a punctuated evolution point of view, right?
Yeah, so I don't think so. This idea that this Big Bang story requires high mutation rate at the beginning, Darrell thinks is true, but if you have 10 to the negative 8 mutation rate, you observe it is not very high. 10 to the negative 8 mutations per division per genome, per nucleotide per division. And in that case, you can simulate that, and the cell side frequency spectrum is exactly that.
Okay.
It's 1 over square, which is actually no solution from channel.
Okay, let's thank Andrew for a very